after meeting many, or all of you, maybe all of you, almost all of you, previously, it's a great pleasure to me to be able to tell you about what I'm doing right now. I really have appreciated the opportunity to participate in this whole workshop. I really appreciated the opportunity to hear about the science that, that some of you are doing, and my friends back there. Yeah, so what I want to tell you about is actually what I'm headed for in my talk is to show you a new interest of mine. And this new interest is causing me to move into a new field, the new field being geobiology. This is a totally new field for me, okay? But I'm going to tell you, and that's because I had an idea, and I want to test that idea about early life on Earth. And to do that, I'm going to have to learn some geobiology. But I need to give you the background of how I got that idea based on the work that we did in my lab at UCLA. So here you see Earth, and life began on Earth about 4 billion years ago. But around 2.3 billion years ago, something happened that could have been a major catastrophe. And it could have been that 2.3 billion years ago, life was extinguished, but it wasn't. And that's something that's always been very interesting to me. And my field is biological inorganic chemistry, and I recently have some ideas about how life may have survived. And that's the story I'm going to tell you. Okay, biological inorganic chemistry. So that's the periodic table, and marked are the elements that are either essential or sometimes essential for in different biological systems. But my story today is, well, there's a bit about iron. Iron's important, but my story today is really about how oxygen almost extinguished life on Earth. Very, very dangerous oxygen. And how I think manganese may have saved it. Okay, so that's what I'm headed for. Okay, so I made the statement that the dioxygen, O2, okay, molecular oxygen, is toxic to all forms of life. Now, you may not think that because we breathe oxygen, we need to breathe oxygen, but it turns out that we have evolved to be able to protect ourselves against the toxicity. So the earliest forms of life appeared on Earth when there was absolutely no O2. And now, of course, the air contains 21% O2. But O2 is a powerful oxidizing agent, okay? It wants electrons very, very much. And living things are full of electrons. So we know that animals, plants, and aerobic bacteria need O2 to produce energy. Nevertheless, all forms of life are constantly being damaged by O2, and life coexists with it either because of evolved antioxidant defense systems or because of repair or replacement of the damage. Okay, so here is our current atmosphere of Earth. Okay, and here is a cartoon that is summarizing the damage over on the top that is done by what are called reactive oxygen species, and reacting with DNA, reacting with membranes, lipids in the membranes, reacting with iron sulfur clusters and enzymes, and reacting with proteins and producing damage in every case. Now, why don't we all burn it up? Why doesn't everything just oxidize? Well, we have evolved some very powerful defense systems. both enzymes like superoxide dismutase, catalase, peroxidase, but also some small molecules like ascorbate, which is vitamin C, or vitamin E, alpha tocopherol. So we've got this wonderful array of protective systems that are in our cells. So, and that is how we survive. Okay, let's go back to the beginning. On the beginning, Earth was very, very different. I like that cartoon, or that little graphic. Not that anybody really knows what it looked like, or maybe Meg does. So we don't know for sure exactly what Earth looked like, but the best guess of early Earth, before life on Earth, was that the gases were similar to the gases that come out of modern volcanoes. And the majority of those are water, nitrogen, hydrogen, carbon dioxide. Okay, but what you'll notice is that there's no O2. There's no O2 in the early atmosphere. There was no reason to have O2. There didn't come out of the outgassing of the Earth. Just no O2, unless you had a little bit of photochemistry from the sun. But it really, O2 on Earth only occurred because of it's a byproduct of oxygen-evolving photosynthesis. So oxygen-evolving photosynthesis, this was something that happened in evolution. Cells learned how to get energy from the sun and to store it in terms of carbohydrates, but what they did was to take CO2 and water, and they produced as a waste product O2. So all of a sudden, you had life that had no oxygen, didn't want oxygen, didn't need oxygen, and all of a sudden, this same life decided to produce oxygen. And that was very dangerous. So the timeline is right here. So 4 billion years ago to the present, and this line, and this line are showing you the pressure, the atmospheric pressure of O2. And you'll see there was none in the beginning. Okay, and this is now current O2 levels. Okay, and the blue one is showing, there's some disagreement about the timing, but oxygenic photosynthesis started right here, and then it's believed that there were little whiffs of oxygen that went up and down. So this is very controversial, exactly how much oxygen when, and that's why the blue stuff is wiggling all over the place. But the point is that 2.3 billion years ago, there was something really huge happened, and the atmosphere went from no O2 to much higher O2. And that time, which was about 2.3 billion years ago, is referred to as the Great Oxidation Event, or the Great Oxidation Catastrophe. So origin of life, and you've got all this time before oxygenic photosynthesis started. Okay, you can see that when oxygenic photosynthesis, the first organisms started producing O2. The O2 didn't rise at first. And the reason it didn't rise is largely because there was a lot of iron 2+, soluble iron 2+ in the ocean. And iron 2+ is very, very, very necessary for life, early life, current life, all life. So the iron 2 reacted with the oxygen and turned into iron 3. And that iron 3, you know as rust, okay? Orange, insoluble rust, okay? So organisms that had learned to eat soluble O2 from the ocean, we're no longer able to do that. So there was this period in here before the atmosphere had a lot of oxygen when soluble iron was becoming scarcer and scarcer. Okay, so soluble iron was becoming insoluble iron. Okay, but then after that happened, then you really had O2, and then you had the reactive oxygen species and all of that stress. So the thing that I've been fascinated with for years was what decided what was going to live and what was going to die right in the beginning. Okay, so the organisms that existed in early Earth were anaerobic. Okay, they lived without oxygen. They didn't need oxygen, and I think we for sure they would have been sensitive to O2. Okay, we don't know exactly what they looked like. They experienced a stress as the levels of O2 and reactive oxygen species go up, and of course, along with that, the iron becoming less and less available. And what happened was that some of them actually managed to find niches where there was very little oxygen. So even now we have anaerobic bacteria that live in various places where it's hard for the oxygen to get there. Okay, so we have modern anaerobic systems, bacteria. Okay, so, but it turns out that they are not a good model for the ancient ones because even they have antioxidant defenses. And there's no reason why you would have had any protection in the ancient anaerobes. So this means a lot of them must have died. And we have no idea what life may have looked like. The only life we know is the life that either became anaerobic or the majority of life that managed to evolve antioxidant defenses and became modern aerobic organisms like us. Okay, so the reactive oxygen species, I'm now going into the background of my research. The reactive oxygen species that's primary, that comes first and does much of the damage of oxidative damage is called superoxide. And superoxide is O2 with another electron. Okay, this is superoxide. And for those of you who are biologically inclined, this is cut of a mitochondria, and this is showing you that normal mitochondrial respiration inside your mitochondria, you are constantly producing superoxide. And that has the potential to do oxidative damage. So I said we evolved antioxidant defenses. The primary antioxidant defense system against superoxide is called an enzyme called superoxide dismutase, and it comes in several different forms. There's a nickel one, a copper zinc one, a manganese one, an iron one. We actually have the copper zinc and the manganese ones, but other organisms have the others. And each of these has a metal, tiny little metal, in this enormous enzyme, which has evolved and optimized to be really good at getting rid of superoxide. And the way it gets rid of superoxide is to make two superoxides react with each other and create O2 and hydrogen peroxide. Okay, this is a reaction that happens spontaneously, but with superoxide dismutase, it happens much, much faster, and there's no time for the superoxide to do any damage. Okay, so I've worked on the manganese SOD, the copper zinc SOD, I've just wondered at how highly evolved they are to do that reactions so well that I've always felt that it was not something that a simple system could do. So we actually wanted to know specifically what damage was being caused by superoxide inside of cells. This is quite a few years ago. And so Edie Gralla in my lab cloned the gene for the copper-zinc superoxide dismutase in the model organism yeast. And the nice thing about yeast, baker's yeast, brewer's yeast, is that the cells are very, very similar to us. And the copper-zinc SOD and so it's very, very similar to higher organisms, so it's a good model system. So Edie cloned the gene for the copper-zinc SOD. The manganese SOD had already been cloned. This enabled her to make yeast strains that were totally lacking in superoxide dismutase. Okay, so what we had then was a cell in which we had removed the major antioxidant systems to protect against superoxide. Okay, from that, we infer that superoxide levels will go up. We expect the cells to be sick, and in fact, they are very sick. And you don't need to look at the details here, but it is clear that they don't grow well. They won't grow at all if you put them in 100% O2, which is not true of regular yeast. Okay, so they're very sensitive to oxygen. They're very sick. Okay, so all of this research had nothing to do with manganese, but one curiosity that other people had observed was that manganese, now not manganese in an enzyme, but plain old ionic manganese, manganese 2+ chloride, nitrate, whatever. Okay, if you add it to some organisms, they will grow better. And also there are some bacteria that have very high levels of manganese. And the manganese seems to be an antioxidant protection system. And so we were curious, okay, exactly how much would manganese help our yeast that had no superoxide dismutase, and would it, how much would it help it? We assumed, actually if you add copper, they do a little better. If you add iron, they do a little better. And this was just curiosity-driven research. We had a little manganese, and they fully recovered from all their ills. Okay, so every single defect that I had on that slide totally went away when we added 5 millimolar of just plain inorganic manganese 2+ salts to the yeast cultures. And it's known that yeast will take up, they also pump out manganese. Okay, so there are only low levels of manganese actually under these conditions, low levels of manganese that are inside the cell. Nevertheless, under these conditions, oh, I don't have that. Okay, you had total rescue. No longer needed a superoxide dismutase enzyme. So that was really startling to me. And because I had always thought you'd need a big enzyme to do that. You know, it didn't make sense to me. It also didn't make sense to me because people were assuming that if manganese was a superoxide dismutase, it would react just the same way that the enzymes react. And the way the enzymes react is to take a metal ion, like copper 2+, and react with superoxide, and give you copper 1+ plus O2, and then another superoxide oxidizes it back, makes hydrogen peroxide. Okay, so they assumed this for manganese, but I'm an inorganic chemist, and I knew that this chemistry was very unlikely. And so I persuaded a student in my lab to actually investigate and see if we could model what might be going on inside of the yeast with manganese and superoxide. And that was Kevin Barnese. Okay, now what was known that confirmed my, well, I mean, it already was known that manganese was not an SOD, okay, which did not catalyze this reaction, at least under the conditions that Cabelli and Bielski studied it in 1984. Okay, but the conditions that they used for that study were not really physiological. The pH was not right, the acidity was not right, and also they had very high concentrations of phosphate ion, and not the kind of concentration that you would have inside the cell. So Diane's a friend of mine, and she's in charge of the pulse radiolysis lab at Brookhaven National Labs. There's Diane right there. And so I said to Diane, are you willing to redo this study, but to change the conditions so that the phosphate level, the pH, everything would be closer to what we would anticipate inside the cell? And she said, sure. And then she's on the East Coast of the US, and I'm on the West Coast. So then Kevin Barnese started commuting back and forth to work in her laboratory. And the reason I had to go to Diane is I wanted to study the direct chemistry of superoxide. And superoxide is pretty unstable, but it turns out that the method of pulse radiolysis will allow you to generate high concentrations of superoxide, and then you can study it. You can follow it spectrophotometrically and so on. And so the pulse radiolytic method that does that, you have to add something else because you need to trap hydroxyl radical, OH dot, not important. There are other radicals that are formed, and you've got to trap them so that the only thing that you form is superoxide. And so what is normally added is either formate or ethanol. And in the old study, formate was used. Okay, well, it turns out that if you have a lot of formate around, to knock it off, and with phosphate, you have to have a lot of phosphate, and that's why the levels were non-physiological. So in pulse radiolysis, people usually think if you put in ethanol, you'll get exactly the same results. But in this case, ethanol allowed us to lower the concentration so that we had levels of manganese and levels of phosphate and acidity that was similar to what you would expect to see inside a cell. And when we did that, we found that the manganese was indeed a superoxide dismutase. Now, this is for chemists. There's Kevin right there, and there's another picture of Diane. This is for chemists, but I'm going to summarize for you in a little graphic what we found, because the mechanism, the way the reaction happens was very surprising to us. It was not like the enzyme. It's okay, so, excuse me. Over on the left is, your left, is the mechanism for the enzymes, which I'm showing you here with copper, okay? And what happens is a superoxide comes in and reacts, and then another superoxide comes in and reacts. Okay, so you see it involves copper 1 or copper 2, and it goes back and forth in a electron shuttle mechanism. The mechanism that Kevin and Diane discovered for using manganese has never been seen in the enzymes, but this is just small little manganese. And the way the reaction goes is a manganese reacts with a superoxide. Manganese reacts with another superoxide. This species is a manganese 3 peroxide. And then those react with each other in a second order reaction. So I think I have it, yeah, a second order reaction, and you get back your manganese 2 and the O2 and hydrogen peroxide. So seeing that again. Okay, so the mechanism was very, very different, but it was effective. And Kevin then modeled, the details of this don't matter, but the point was to figure out, if I take the superoxide dismutase enzyme outside of the cell, how much manganese do I need to put in there in order to have the same effect? And the result of this that he found was concentrations of manganese that are very easy to reach in vivo. So this meant low concentrations of manganese inside the cell could in theory substitute for the enzymes. And so we then we studied the manganese phosphate, we studied the manganese bicarbonate. We also looked at concentrations of phosphate, bicarbonate, and manganese in the cell. And there's no question that in theory, okay, it's hard to know exactly what's happening inside the cell, but there's no question that in theory, the manganese, there's enough manganese and enough phosphate and bicarbonate around so that you could substitute for the enzyme. And we think it's very likely that the bacteria that protect themselves with manganese are using this mechanism, and we don't know whether higher order cells, whether our cells do that. Okay, nobody's ever asked that question because they didn't know whether they didn't know that manganese itself could be a superoxide dismutase. So that's what I was focused on when I had this idea that led me to geobiology. Okay, so this idea that in addition to the superoxide dismutase enzymes, just plain old superoxide could protect against superoxide toxicity. So I learned, I went to a meeting, a scientific meeting, and I presented this work. And a geobiologist asked me a question, and he said, and in the context of that question, he said that the levels of manganese in the early oceans were much higher than they are now. Okay, so it was that question, that discussion at that little meeting that started me on the pathway to changing fields, I guess, whatever I'm doing. Anyway, so for sure, it is now established that that low molecular weight manganese, phosphate bicarbonate, and actually some other ligands can act in place of superoxide dismutase enzymes inside of cells. And that probably functioned early, in early life, right? Because manganese was abundant in the early oceans, and so you get manganese inside the cell, it may have been protecting the cells. But that doesn't really explain, let's see if I, okay, that doesn't really explain about the earliest life, because it's not just superoxide that's the problem. The biggest problem in the beginning would have been O2 itself. Okay, these are the reactive oxygen species we worry about now, and here are the defense systems. Early life, none of these defense systems, and O2 itself would have attacked all of these species. And so O2 itself would have been a lethal toxin. So how do you protect against O2 before it even turns into superoxide? Okay, well, what I thought about for a number of years actually was that it was interesting that the stress that you had first was too little iron. Okay, you didn't have enough, the iron was disappearing in this early stage. And we know how cells evolve to deal with that. And one of the principal ways they dealt with it was, okay, here, I meant to tell you, this is how the geobiologists figure out the timing of the oxygen going up. These are banded iron formations. That orange is rust. And that's why they know that the oxygen went up and down, okay, with these different layers that are formed in the rocks. Okay, so what we know is that there are, in modern cells, in many modern cells, bacteria, yeast, so on, ferric reductases. These are enzymes that sit in the membrane of the cell, and they take electrons from the inside of the cell, and they react with insoluble iron 3. Okay, so maybe they're sitting on a piece of rust, and they feed an electron to it, solubilize it, and then they pump it into the cell. Okay, we have that enzyme is very common in the membranes of cells. And the thing that really fascinated me was the principal class of those enzymes, which are called ferric reductases. This is the structure of the ferric reductase sitting in a membrane, right? It's taking electrons from the inside of the cell and putting it on the outside where it takes the iron 3 and solubilizes it. But it turns out this is the same family of enzymes as the NADPH oxidases. And the NADPH oxidases are modern enzymes that we've got in all cells that do the same thing. They take electrons, but they produce superoxide. It's used for signaling, it's used for defense against bacteria and so on. But it's essentially the same enzyme. Okay, so this is what led me to my idea. So if the earliest life, if the first thing, lack of iron, was the stress, then the cells figured out how to take electrons from the inside of the cell, move it to the outside of the cell, and get their iron. If those same systems, whatever they looked like in their primitive form, could take the O2 and react it and make it into superoxide, then it was a way to keep O2 out of the early cells, because O2 is hydrophobic. O2 goes through membranes very readily, okay? But O2 minus is hydrophilic. It's water soluble, it likes water, it doesn't like membranes, it doesn't like lipids, and it's not going to go inside the cell. So my hypothesis, which sounds very simple, but I'm told by the geobiologists is new, is that the earliest cells protected themselves against O2 by reducing it, or by taking O2 and making it into superoxide and keeping it from going inside. This would then buy time for the evolution of other antioxidant systems. Okay, so my idea that manganese ion, and then you can deal with a superoxide outside of the cell and protect from the extracellular damage because you've got these high levels of manganese in the early ocean. Okay, so my hypothesis is twofold. One, that O2 was being reduced to O2 minus on the exterior of cells in order to protect the earliest cells, using the same systems as reduce iron, and the other part of my hypothesis is that manganese then took care of the superoxide, at first outside of the cell, ultimately probably inside of the cell, before you had the really advanced superoxide dismutase enzymes. Okay, so we still don't know what, I wish you could figure out what was alive and what died. But I think that the cells that did survive were able to throw off electrons on O2. And this was very interesting to me because 2 years ago there was a major paper in Science in which it was reported that bacteria many different kinds of bacteria in the oceans. It was known that they produced superoxide, but it was thought that that was photochemistry. But what was published in this Science paper 2 years ago is, no, it's not just photochemistry. Even if you go into the dark, all of these bacteria are constantly making superoxide, and nobody knows why. Okay? These bacteria, there's a steady state level of superoxide in the oceans generated by bacteria that are producing extracellular superoxide. Now, I think this is a remnant of this early selection protecting them from O2. This would be consistent with my ideas about what may have happened in early life. Oh, this is just to show you. This is a log scale. These are different. Here you see iron. This is time. Okay, 4 billion years. Present, you see the iron dropping as it becomes insoluble. It comes out of the ocean. And here's the manganese, which was substantially higher than it is now. Okay, so what am I going to do? I want to find out if this was what happened. And in order to figure out what happened in earliest life, really the only evidence you have is the geological record. And so the earliest cells that produced O2, I figure that those cells that first produced O2, they got energy from the sun by photochemistry. They had O2 as a waste product, right? Our best guess about what they look like would be modern cyanobacteria. These are bacteria that photosynthesize, very similar to the way plants photosynthesize, okay? And they're really pretty color, which is why they're called cyanobacteria. And this is a great picture I pulled off the web from a great website, the astrobiology.net website. You can see cyanobacteria. This is a close-up of cyanobacteria, and that's a bubble of oxygen that it's producing. Okay, so imagine the early life, and you have cyanobacteria, and they're generating this incredibly toxic gas. Well, cyanobacteria tend to live in mats, and in the ocean and in shallow water and so on. And these mats form stromatolites. Okay, this is stromatolites formed today, built by colonies of microbes. And microbes such as cyanobacteria create these structures. And you can go to places where you can get rocks that go back to the time of early life on Earth, and then look at what is there and try and deduce what may have happened. Now, I don't know how to do that. Okay, so but I want to learn. Okay, and so I'm going to be, next year, I'm going to be visiting associate at Caltech, associated with Woody Fisher, who's a geobiologist who specializes in the transition to O2 atmosphere and also in manganese. So he turned out to be a perfect match, okay? And so that's my new direction that's going to start this September. And I'm very excited about it. So maybe someday I'll tell you if I got evidence to support my hypothesis. But I do want to thank, that's where the story is. I don't have the answer. But I want to thank a lot of people. I mentioned Kevin, who did the pulse radiolysis. Raylene Sanchez did the yeast work with no SOD. Edie Gralla is my longtime collaborator. I'm an inorganic chemist. She taught me all the biology I know. We've worked together for many, many years. Diane Cabelli, wonderful friend and collaborator. Woody Fisher, new collaborator, who says my ideas are new, and which I like. And I also want to thank the laboratory of Professor Wanu Nam, who is my former PhD student, who put together a grant, a world-class university grant, that brought me and Ken Carlin and Shanfu Kazumi to the lab with these wonderful students. I'm sorry, you guys are not in this because it's an old picture. It's a wonderful laboratory where we collaborated and talked about science all the time. And many of the ideas that I talk about today, I developed while I was talking with these wonderful people at IWA. And it was a really great experience. I want to acknowledge them too. And I want to thank you for your attention.